What I hope to do in the next hour, hour and a half is walk you through a lot of information looking at emerging therapies in lymphoma in 2014. And I too see a couple of familiar faces out there, so that's excellent. And I've also realized that I know how to shorten my bio now. That'll make it a lot easier. So I treat lymphoma, and that's the thing that I'm the most interested in doing. And in our group, we're lucky because we can do pretty much everything around lymphoma. So we give primary therapy, we do relapse treatment, stem cell transplant work, and novel drug development. And I think that gives us a good opportunity to sort of have a good landscape on which to look at these sorts of topics as how treatments may be changing things in lymphoma. I think it's important for you guys to realize as we get into this place, one needs to work with a lot of companies in order to be able to do these studies. And so you should always be cautious when you see us present data from these types of research studies. Admittedly, I work with a lot of companies, so I think that allows me to say that I don't hold one of them, I don't like one more than any other. And yeah, I didn't grow up in Toronto, but close, and my loyalty has always been to be home. All right, so three objectives essentially. So what I hope to do is to provide some context in the beginning so that you can understand some of the emerging therapies in lymphoma. I'm going to quickly talk a little bit about clinical trials because I think that is important. Certainly the patients and the families out there around lymphoma, this is something you'll hopefully be confronted with because we're doing treatment studies often in the frontline setting where we still hope to make things better, and it is important to have options available all along the way. And the last part of this I've always found to be a bit of a challenging thing to do in terms of a presentation, which is how do you run through the laundry list of all of the new drugs that are out there in a way that makes sense and kind of keeps it interesting. So we'll try and do that a little bit in the context of the drug as well as the disease. And so that may help either answer some of the questions or leave them a lot open because you've probably heard of a lot of promising drugs, but there may not be data yet or good enough data in the disease that you're interested in, okay? And that becomes important for context. So this was the state of the art in lymphoma essentially from 20 years ago. And this is an update of that publication. And what this looked at was a variety of different standard chemotherapy drugs that were given in combination with the idea of building on CHOP. So that was the standard chemotherapy in lymphoma back in the old days. And people had thought that adding more standard drugs to that regimen would make a difference. And things looked very encouraging. But in retrospect, what we learned was that standard chemotherapy had very little to add. And so this is looking at the proportion of people that are alive who had gotten one of those 4 regimens. And what you can see, there's up to 8 drugs in one of them. CHOP was 4. It was the easiest to give, and they all performed identically. Okay, so my conclusion from this, as many concluded, was that standard drugs will get you so much, and then beyond that, you're going to have to look to build with a different platform. It's always good to have a very good history lesson as a story to tell. And so I'm sure all of you, or most of you, will be familiar with this drug, rituximab. Again, the patients, probably a lot of you have received this in the setting of B-cell malignancies. And so what is rituximab? Okay, so it's a monoclonal antibody, so that's something we're going to talk a bit more about. So antibodies, the idea for this, biotech companies really stole from the body, right? So your body makes antibodies, which are proteins that help you fight infections. And so why not make that type of a targeted treatment against a cancer? And so what you need is a target. So in a virus or a bacteria, it might be something specific to the virus or bacteria. And then your body's immune system recognizes it, creates an antibody, and the antibody helps to kill the bacteria. So that was the principle, and this got engineered in a lab. And the target was something called CD20, which is a marker that is found on almost all B-cell lymphomas and is actually found on normal B lymphocytes in your body. And so these are part of your immune system that help you fight infection. And it's found on the surface of the cell, which means it's a great thing to attack because if you have this antibody, the drug floating through the bloodstream, it will eventually see the target on the cancer cell and it will attach to it. And then it does what it does. And so just to put things in context, this was the original study that looked at the effect of rituximab in indolent non-Hodgkin's lymphoma. So this was basically in a group of patients with relapsed or refractory follicular lymphoma who'd gotten traditional chemotherapy because that's all we had back in the 90s. And the thing that stands out is if you look at here in the middle, the response rate, so what proportion of people had their lymphoma shrink by 50%, and that's how we generally assess how a drug works, was about 50%. Okay, so half the patients derived significant benefit from this in terms of having their lymphoma shrink. How long did the lymphoma stay shrunk? So the duration of response was just over a year, so 13 months. And that, if you can imagine, was a phenomenal finding for a single drug. Okay? And the other important thing, how was it to give? What was the toxicity like in terms of the patients that got it? So it was actually pretty easy to give beyond, as you know, some of you may have experienced, when you get the drug intravenously, you occasionally can see some side effects as the drug runs through the intravenous, mild allergic reactions typically. And so I want you to remember this because this is the bar, this is what I think is a very high bar that established a drug that looked very good in lymphoma. And then what happened? Well, a host of studies came out, and this is one of them. This is the original French study that looked at combining rituximab with CHOP in people over the age of 60. And unlike that other slide that I showed you where there was no difference in survival with 8 drugs, standard drugs versus 4, you can see here there's a huge benefit in terms of improving survival, curing the disease. Okay, and that's why rituximab became a standard treatment in aggressive histology lymphoma. And this was replicated in many other kinds of B-cell lymphomas and essentially confirmed this in all those other diseases. And this is why this is part of standard treatment for essentially all B-cell lymphomas. What was the other story there? Well, clearly, research can get translated to patients and improve outcome. And here, there's a good success story in lymphoma in that, again, treatments have improved outcome, treatments have improved how patients can tolerate treatments, and this is all the result of a lot of painstaking work, a lot of engagement with patients, a lot of investigators participating, and so there's a lot of success that we can take from research in the field. Importantly, this has also led to change not just in the clinical trial, but changes that happen with the day-to-day care of patients moving forward. The important thing in all of this, you know, if your approach with going on to a clinical trial, you know, I've just shown you an example of a huge win, but what if you were in the study before and it wasn't a win? Okay, and that's one of the things we have to be aware of. So who actually benefits at the end of the day from participating in a research study? It's not always the patient who's in the study. Okay, if you're lucky and you get onto a good study and in retrospect you know that it worked, sure. But otherwise, the people who ultimately benefit are the greater community, right? So all of the people, all of the patients who have the disease will learn from people going through these studies whether it's actually helped or not. And if it was a randomized study where half the patients get the new treatment and half don't, if the treatment worked, the people that got it will benefit. The people that didn't, you know, unfortunately won't. But when this is an unknown, these are, you know, these are the rules that you kind of have to sign up for to participate in. Some of the studies do allow that when you know the drug works, this idea of crossover. And so if you didn't get the experimental treatment and it looked to be beneficial, you could get it after the primary treatment didn't work. And so that's one of the ways that we try to at least provide some benefit for other people when they go into studies. So a couple of quick points now just to try and highlight a couple of differences about the types of lymphoma research that we do. So one of the areas that we do focus in is something called translational research. You've probably read a lot about that or heard about it. And the idea here is we're trying to translate discoveries either from the clinic back into the lab or vice versa. And so the idea there, again, one of the research colleagues that I work with, you know, does a lot of stuff with test tubes and cultures and animal models. And when you develop a drug in that area and it looks very promising, again, that's the scientific basis to then ask the question in a group of patients. Similarly, if we made an observation that a drug works well in breast cancer, what if it worked in lymphoma? And can you show that there's a relationship between the target maybe not a breast cancer gene, but a tumor gene that may be involved in different cancers, take it back to the lab, see if it works there first before you bring it back into the clinic. And so typically, most big academic research practices will try to have a couple of different ways of trying to shuttle back and forth between those two areas. And so as a patient, what can you be asked for? Outside of participating in a study with drugs, we may ask for pieces of tumor from biopsies that have already been taken so that we can study them in the lab. We may ask for biopsies to be collected on treatment to look at how the tumor responds to treatment. We may also ask for blood so that we can look at the effect on normal blood cells or on other markers, even the immune system or what have you, to try and see if these treatments may be doing other things. Okay, and so it's very important. The studies aren't always about drugs, though the ones that we tend to show you, particularly as we move forward and the topic is emerging therapies, is very much about drugs, but there are a lot of other things that happen in the background. So we talk about clinical trials in terms of the phase of the study, and I think this is another important concept to be aware of. And again, very simplistically, these were numbered from phase 1 to 4. And the type of question that's being asked in the study is reflected in the phase of the study. And so we'll just start at the top there. So phase 1 studies are often the first time that a drug has ever been given to a human. They're also potentially the first time 2 drugs have been given in combination together. And so the purpose of that study actually has nothing to do with determining how effective the drugs are. The purpose is actually to see how safe the drug is, or how safe the combination is, and to understand how frequent the side effects are. Okay, so when we're offering someone a phase 1 study, that's often because there may not be other options that are available, or because, again, the combination looks so promising because of the 2 drugs' single agent activity that it would be a great idea to do this, whether it's we're taking a standard treatment in the frontline setting and we're adding a new drug to it. Okay, so those are probably the most risky types of trials that are here because we have no idea if they're going to work. We often start at very low doses that may not be effective because we don't understand what the side effects may be. Okay, phase 2 studies are like the study with rituximab that I just showed you. So typically a larger study, we give the drug, we have an idea that we know the dose, and we know the types of side effects we may expect to see. But what we hope to see here is understand those side effects a bit better, and then to really get a sense of how well the drug works. Okay? And often when you're looking at patients with relapsed lymphomas, phase 2 types of studies are often what we're talking about because there are often a host of these drugs available, and we're trying to see if a promising drug looks good enough to be taken to the ultimate test. And that's what a phase 3 trial is the ultimate type of study where you have a defined standard treatment. So again, that was the first study that I showed with CHOP. And then comparing that with CHOP with rituximab, where you have a treatment that looks promising, and if it's better than the standard of care, we'll replace it. Okay? And that's the type of study that to a patient is actually the lowest risk because you know that at a minimum you're going to get the standard treatment, and there's usually a 50-50 chance that you're going to get an experimental treatment that we hope will be better. Again, the difficult thing there is it's a hope, we don't know. So often the study ends up being it's just as good, but admittedly occasionally these trials have been done, and it's whether the treatment ends up having more side effects or not that it makes it actually a less effective treatment for whatever reason. That does happen occasionally, but we try to weed those out along the way earlier. And then lastly, there's what's called phase 4. It's kind of more like understanding what happens with a treatment at a population-based level. So this is where you may see rare toxicities come out. You may understand side effects that happen at 5, 8, or 10 years after people get treatment. And this is important for the vigilance both of the company and of the community that treats the disease, so we can understand the impact of our treatment outside of just the types of studies that we've done with a few hundred patients. And all the way through this, just as a reminder, as I've noted at the bottom there, none of this has to do about anything with an individual patient, right? This is about proving the treatment. And that's one of the hardest things here because Clinical trials sound very exciting. Investigators get very excited talking about them. But at the end of the day, the purpose of the study is really not to benefit one person, right? And so that makes it difficult, but you may benefit. An individual patient may benefit from this, but again, the primary reason to want to do this is more about the discovery and helping, though admittedly, if the drugs work, they will be a very good choice for someone who may need them. Okay, but that's the push and pull of doing clinical research. So we're going to look at now some examples of these treatments, which are emerging to some extent or not, and talk a little bit about their place and where they're going to go. And this is going to build on the story of the chemotherapy that I mentioned in the beginning. And this is actually then a chemotherapy question. So this was the pivotal or most important study that looked at this drug called bendamustine, which again, you've probably heard something about. So bendamustine is a traditional chemotherapy drug. Despite a lot of buzz about it being a new drug, it's been around for decades. It was developed in Eastern Europe, and there was a lot of experience behind using this drug in Germany. And so what happened, again, a bunch of investigators in Germany chose to do a study where they looked at indolent lymphomas, again, typically, most commonly follicular lymphoma, but there was a smattering of other diseases, including mantle cell lymphoma in this study. And then the question that was asked was, with rituximab as the backbone, because we know rituximab is the drug in this disease, what was the best chemotherapy partner to give with rituximab? And so they compared their drug, bendamustine, versus CHOP. And CHOP we had a lot of familiarity with, and the bendamustine less so across the world. And one important point, the bendamustine as it's given is given as a 2-day treatment, day 1 and day 2. The rituximab you can get day 1. The CHOP rituximab, as anyone knows who's had it before, is a 1-day treatment for all of the intravenous chemotherapy, and you get pills, prednisone, for 5 days in total, which you can get at home. So when you look at the study, what this shows is equivalence, okay? So the response rate, so the proportion of people that have the lymphoma shrink by at least 50% was exactly the same. But what was interesting, there was an improvement in what was called complete remission. And so that's essentially making your CT scans look normal. Okay, so 10% better here. And just very quickly, what these p-values represent is when you do the math and statistics, is that difference actually real? Okay, and it's not potentially through chance. And so when that number drops below 0.05, that means it's entering the realm of what we believe statistically it's likely that you're seeing a real effect. Okay? And so this is important because actually the purpose of the study was to show bendamustine was just as good as CHOP, not that it was better. And what they actually ended up seeing was that it was better. And so progression-free survival, so essentially this means the time that the lymphoma stays controlled until it progresses or grows was noticeably better for the patients that got bendamustine in the red curve compared to those in the blue curve. And when you look at the numbers here, the median, so half of the patients were still in remission at 70 months with the bendamustine combination compared to only 30. Okay, so it appears to make people's disease shrink more or better, and it keeps you in remission longer, right? So that's a pretty big finding. Unfortunately, it didn't show, so there are two curves there, they totally overlap, so it doesn't matter which one's which, but it didn't actually show that people lived longer when you got the Benda, despite staying in remission a lot longer. And so this is one of the tough things we struggle about in these studies. Why did that happen? And I can give you some theories, but I don't know the answer for sure. One of the theories certainly could be that phenomenon of what I talked about earlier, crossover. So if you got the CHOP as part of your treatment in the beginning, once people saw this drug looked really good, what do you think the patients might have gotten as the second treatment? Bendamustine, right? And if the drug's still really good, that may make up some of that difference. That's the important thing in diseases like indolent lymphoma, where the trajectory is long and you have opportunities to treat people again over time. A treatment that's effective up here, but you didn't get it, may still be effective down here. And you may make up some, you may not make up all of that difference, but that may be part of it. And there were other imbalances in how the people got treated second line, that's part of it. Or part of it may just be this treatment doesn't affect survival, we don't know. And the study, to be fair, was not designed to look at survival in follicular lymphoma. That's becoming a tougher thing for people to do because the drugs now, the treatments are so effective, you'd need to do studies that look at 10 or 15 years, and that's often beyond the patience of a pharma company or beyond the patience of the investigators, particularly if they're nearing the end of their career. The other point about this study that I want to highlight that's a huge win, which is the main reason that this drug got into this type of study. So this looks at toxicity, and I'm just going to highlight those top three columns, top three rows in this column on the right. So these are people and looking at the degree of how badly their blood counts dropped during treatment. And we worry about fever and neutropenia, we worry about bleeding, we worry about anemia, maybe the need for transfusion. And in all of those accounts, the CHOP caused more side effects, was worse on blood counts than bendamustine. So now you have a treatment that is more effective and easier on patients, right? So clearly a win. And similarly here, in terms of the side effects that people notice, not just the numeric side effects, you can see here, bendamustine doesn't cause hair loss, CHOP does. Far less likely to cause numbness and tingling in your hands and your feet. So neuropathy symptoms, far less tough on the mouth in terms of side effects, far less likely to lead to infectious complications. The trade-off was that you do see more skin reactions. And so people, if you have to ask me, what is the most common reaction you see with people getting bendamustine? It's actually allergic skin reactions. That doesn't prevent us from giving the treatment. It may be something you can treat with antihistamines that you can get over the counter or a bit of a steroid cream or something like that. But I think when you look at the differences there, it's still a win for patients. So I'm going to show you another study looking at bendamustine, and this is a trial in CLL actually. And so the idea here was fludarabine, cyclophosphamide, and rituximab was the standard. So that's the equivalent of CHOP or CVP in terms of what we were giving in lymphoma. And they were trying to compare that against bendamustine based on the data that I just showed you, which was the idea that this was a drug that was more effective but had fewer side effects. Okay, and they were trying to do this as a primary treatment, so the first treatment people were getting for CLL. And so what they ended up doing is they did the same sort of randomized studies. This is a big phase 3 trial where half the people went on to get the FCR and half the people went on to get the BR. And so this becomes important because CLL now we're thinking about in a slightly different way than we did 10 or 20 years ago. So there are treatments that are more aggressive, and so they're reserving these for patients that are fitter, typically younger with not a lot of other medical issues. And this was aimed specifically at that group of people. And what they were also trying to show here was much like what they were trying to do in that previous study. Not to show that bendamustine was better, but that it was just as good. Because they believed that if you were just as good and it caused less side effects, it was a clear win. And so what ended up happening, unlike the study that I just showed you, here the response rate, so the proportion of people that had their tumor shrink by a half, was actually lower for the people with bendamustine, at least in terms of achieving a complete remission. So the people that had the disease shrink by at least half was exactly the same, but that deeper response, making things entirely normalized, you saw more commonly with the standard treatment, which was this fludarabine, cyclophosphamide, rituximab. What did that translate into? So when you look again at that time, the people, how long do they stay in remission, or how long does it take until they progress? What you can see here is that the better curve is actually with the fludarabine arm, not with the bendamustine arm. And the difference there is statistically significant, showing that the patients that got the standard treatment did a bit better. Again, in terms of overall survival, looks exactly the same. So again, it comes back to that point with the survival. What happened? Did people get switched over to either treatment in between? Were they treated differently afterwards? Again, the study also wasn't designed to look at the question fully in terms of overall survival. Just to highlight here, the type of treatment that you got mattered. And so they looked at all these other factors in terms of the balancing points, in terms of how people did. And in general, the treatment was very important. The other thing I'm going to highlight here is that age was also important in terms of how you did with regards to treatment, not surprisingly. So above the age of 65, it's a little harder to treat people with CLL successfully. On the toxicity side, though, they did confirm what was seen in the other study, which was that in terms of really bad side effects, they were far less likely to happen if you got bendamustine. So again, here we're seeing a slightly different story. The treatment was better in terms of how patients tolerated, but there was a trade-off in terms of how effective it was. And importantly, as you can see here, infection in CLL is a big concern. And it was, about 15% less likely to happen in the bendamustine arm. And now you get to see potentially that there could be a perception of choice, okay? So, the conclusions, the groups weren't entirely balanced in between the two treatments as far as some of the risk factors with treatment, but FCR looked better. FCR did come with a price of higher toxicity. And what's the take-home about this? I don't doubt that bendamustine is a less toxic treatment than many of the standard things that we give, so all of these studies show that. In Canada, we weren't giving CHOP a lot in follicular lymphoma because it was a more toxic treatment to begin with, and we were giving a slightly easier treatment, which is called CVP, and we might save the CHOP to give later in the disease course. And so it's a little difficult for us to make that comparison if our standard would have been less toxic than bendamustine. I think I'd probably say it would probably end up being about the same, okay? But if this was better, likely in terms of keeping people in remission longer, and the side effect profile was still good, we took that as a win, which is why it became a standard treatment in follicular lymphoma or other indolent lymphomas. And in terms of CLL, the take-home has been, it's hard to really know what to do about that study. It's good in one way, it's less good in another, and we'll need to see what's going to happen there. But the importance of that interpretation becomes critical, because if this drug is going to be approved in Ontario, if it's going to be approved in Canada, based on that data, it isn't a slam dunk, right? Because you can see there's trade-offs there in terms of what happens. But it's an important option, and it's certainly something that clearly has a role in CLL. It will just be trying to tease out those details a bit better. So this is another frontline study in CLL about another new, and I'd say potentially very exciting drug, a drug called obinutuzumab. I'm probably just going to end up calling it GA101 because that's what I've been so long used to calling it. So this was given in combination, again, with another chemotherapy, chlorambucil, and it was being compared against rituximab. So this is another antibody treatment like rituximab. And this was done also in CLL, also in primary treatment. But unlike the other study, the main difference here is this was looking at people that were older or had other medical problems. So the group of patients that wouldn't have gotten into the study that we just talked about. So this is some fancy structural chemistry that shows what this drug looks like. And essentially, the way I think to easily think about this is kind of like a second generation rituximab, if you like. And it's been engineered in a way that it works differently. And, not to, diminish 10 years of research or whatever, let's essentially just leave it with the idea that in the lab, it looked better, okay? So it killed cancer cells better, and it looked promising enough that people would try and take it ultimately into a large study in patients. And the mechanisms, again, are listed here. The important thing, one of the difficult things with rituximab was to show that it actually directly killed cells. This drug directly kills cells, and this is potentially a reason to believe it would be more effective in diseases where rituximab was less effective. And so what they did in this study was they tried to get at two questions in this older, less fit group of patients with CLL, where again in Canada, the standard was to do this, to give this drug called chlorambucil. It's a pill, very non-toxic, very simple to give, and so in people who had other medical issues, perfectly reasonable. What they tried to do was to potentially show that rituximab was better in combination, because that hadn't been shown before. We all believed it was going to be the case, but then to look at the new antibody as well and to see. And the question then was even, were these better than chlorambucil alone? And then was there a chance that one of the antibodies was actually better than the other? Very big study, 800 patients. And again, in terms of the patient benefit point, here's an interesting thing. So if you went into this study, again, this was a study run in Germany, it was allocated in a slightly unusual way in that you had a 1 in 5 chance of getting the chlorambucil, and then you had a 2 in 5 chance of getting the rituximab or the GA101. And that provided some incentive to the patients then to say, you know, it looked like you could have a better chance to get one of the new therapies. Scientifically, it was important to them because they needed less patients to show the benefit over chlorambucil, and they needed more patients to show the benefit versus the two drugs, the two antibodies. But again, an interesting example to see where the design allowed potentially to help patients more. This trial got a lot of buzz when it was presented at the American Society of Hematology meeting back in December. And that's because this was the second treatment to show an improvement in overall survival for patients with CLL. And that's shown here. So if you got the new antibody GA101 compared to the chlorambucil, which was the standard treatment, the survival improved. Okay? And that was not, maybe that was what we all hoped to see, but that was a little bit better than what they'd actually designed the study to show. When they then looked at the two antibody arms, so the GA101 versus the first generation drug rituximab, there was no difference in survival, though again, you'll remember what I told you about the statistics here. If this number was below 0.05, we would have said there was a statistically significant difference there. And so you can see it's 0.08, close. We'll have to see with some more time, because this is early follow-up of these studies that we're now seeing, if that gets better and we see the real difference, or if it even gets smaller because maybe some of the patients are going to go on to get GA101 if they got rituximab, or they're going to get other treatments. We don't know. So what can we conclude from that study? So GA101 or obinutuzumab appears to be a better anti-CD20 antibody than rituximab in CLL. So what I didn't show you was the staying in remission part of this was better for GA101 versus rituximab, even though that didn't translate into the survival difference yet. We're very excited about the drug. The company that makes the drug, Hoffmann-La Roche, obviously is very excited about the drug. And they're now doing what you can all probably guess we would want them to do, is they're replicating those studies in diffuse large cell lymphoma and in follicular lymphoma to try and show proof of concept that a new antibody may be better than the standard. You know, when did the rituximab story begin? So that initial publication that I showed you was 1998. So how long is that now? You know, almost, well, 15 to 20 years later, we're now seeing the second generation drug. So it is exciting, but the unknown here, again, to be a bit strict about it, we don't know if the success in CLL is going to translate well to these other diseases, and it becomes incumbent on us to prove that that's the case, okay? So obinutuzumab at the moment is now sort of working its way through regulatory approval in Canada at Health Canada, and then we'll have to see at some point when it comes to the provinces and nationally in terms of getting it funded. So here's another one of those anti-CD20 antibodies. So again, another cousin of rituximab or obinutuzumab, and this is a drug called ofatumumab. And so again, it targets CD20. This is another description of the target in the cancer cell membrane. The difference here is that rituximab binds CD20 over here, and this drug binds CD20 over here. And what that led to, again, preclinically in the lab, were some differences that suggested that this drug would be better than rituximab. And they did a pilot study in CLL, because CLL was the disease out of all the lymphomas where rituximab appeared to have the least benefit. And what they showed was that ofatumumab looked better than patients who had than what rituximab looked by itself in CLL. And so the study that led to ofatumumab's approval by the FDA in the US was this study. And they took, so this was a large phase 2 study. So again, there was no comparison. This is just patients getting the drug. And they looked at a bad group of patients with CLL. So CLL patients that were refractory to 2 standard treatments that we would give, fludarabine or a drug called alemtuzumab, also known as Campath, or they took people with very bulky disease, so people who had large, so at least 5 centimeters or more, areas of CLL and had also been refractory to fludarabine. And what they showed in that study was this. And as you can see here, the response rates look very promising, but again, this isn't compared to any other treatment. This is just what you can conclude about this on its own. So the response rate was 60% here in the double refractory patients, and in the bulky patients was about 50%. And remember, I told you, a very high bar is to say your drug works 50% of the time. And then how did people do? Well, so compared to those other curves that I was showing you about survival, or how long people stay in remission, the median in this case was about 6 months. Okay, I will tell you that was better than what you would likely have been able to achieve with anything else. Okay, but this wasn't compared against anything else. And the survival, again, was decent at, again, a bit better than a year. But the difficult thing then becomes, how does it look in terms of toxicity? So the toxicity here, when we look at grade 1 or 2, these things are often quite mild and you don't need to do very much about it. Grade 3 or 4, when you see more side effects. And so the red here is grade 4. And so those are significant medical side effects that would typically require some sort of medical treatment. And this just looks at people, how they reacted potentially to the drug. And again, with all of these anti-CD20 antibodies, a common thing is that when you're getting the drug infused, you can see allergic reactions. And a grade 4 allergic reaction would be quite serious. But they were very uncommon. Again, the vast majority were grade 1 or grade 2, which we can typically manage quite easily with things like Tylenol or Benadryl or other antihistamines. What you saw here is that in terms of infection rates, again, you would see that in about 25% of patients, and it did have some consequence in terms of blood counts, but it was generally not that bad a drug to give, okay? But again, hard to know in the absence of a comparison. So the reason that I've left this blank is that, again, at the American Society of Hematology meeting back in December, there was a phase 3 presentation looking again at patients with older CLL, and this was a randomized study comparing this drug to chlorambucil, where they actually saw a big improvement again in terms of progression-free survival. And this is now a drug that will be sort of working its way again through that approval pathway for the treatment of CLL. Okay, so again, another drug to have available in CLL. This was confirmed in a frontline study, so primary treatment against a standard treatment, which is chlorambucil, but unfortunately I was unable to get you any real slides from that one yet to show. So ofatumumab clearly has a role. I think the question for us here is if you start to say, I need my first treatment for CLL, I'm not, I'm a little older, I have some other medical issues, what's the right treatment? And this is where the studies don't answer the question directly, because what would you like to do? I'd love to compare this one against the other one that I just showed you. Has that been done? No. And so then how do you do that? You get into this dangerous game we all play where we look at the results of this study and we're going to compare them mentally against this study. And it's very difficult because the patients obviously weren't the same. They may be similar, but they weren't the same. And outside of CLL, there haven't been compelling studies to show that ofatumumab offers too much beyond rituximab, and that's probably why it's still been unfunded. No one sort of pursued that in non-Hodgkin's lymphoma in Canada. So again, it'll be an interesting time to see now with 3 different drugs that have the same target, how these things play out together. But the difficulty again, as you look to try and use these things for patients in the clinic, is that the head-to-head studies haven't been done. And most importantly, the funding for some of these agents we're still waiting for, though the data are as of quite new in the past 6 months or so. Okay, so that is going to conclude most of the antibody part of what we're going to talk about. And the area that people have focused a lot on is now something called B-cell receptor signaling. And what this means, okay, so again, B cells, as we talked about, may be normal B lymphocytes in your body, but the majority of lymphomas that we see are actually B-cell lymphomas. And so the principle here is that there's something called the B-cell receptor, which is over here. And what ends up happening in a number of different types of lymphomas, this includes CLL, it includes mantle cell lymphoma, it includes subsets of the other types of lymphoma, large cell lymphoma, follicular lymphoma. The B-cell receptor is chronically turned on, okay? And nothing in the body should ever be turned off and not turned on and not turned off, okay? So something in the cancer cell has gone wrong that allows this pathway to stay on all the time, and this leads to uncontrolled cell growth, which again is a hallmark of cancer. So there are a number of different targets in this pathway, and I'm going to highlight a couple of them. So this drug called ibrutinib, which targets something called BTK, which again is an enzyme downstream from the B-cell receptor, and something called PI3 kinase, or PI3K, which is targeted by a drug called idelalisib, which is also known as GS1101 before it got that name that's a little hard for people to pronounce. So there are a number of other places that you could start to target this, and there are drugs for these other areas, or multiple drugs that may be in this area. And so this is a very exciting place and time because, again, a whole host of new drugs and a pathway that you think you understand, and it seems to be important in lymphoma. And so there was a lot of excitement about this study because it was presented in the New England Journal of Medicine last year. And this looked at that first drug called ibrutinib, again, in people with CLL. Why am I spending a lot of time on CLL? It's probably a good question. So it is a type of lymphoma, but in terms of where a lot of the new drugs have had their initial studies read out, it's actually been in CLL. So it's made CLL a very busy area for new drug development. The thing that's, I think, part of the reason this has happened is because the bar has gotten so high in terms of what's happened in other non-Hodgkin's lymphoma like follicular and DLBCL. This has kind of been the area where companies have invested time and effort because second-line treatment in CLL, unfortunately, there weren't a lot of very good treatments up until very recently. And so, everyone realizes there's an unmet need. There's also an opportunity to develop a drug for a company in an area of unmet need, and it made a lot of sense. But, trust me, the principles that are here are going to apply very broadly to what we see in lymphoma, and we hope some of these results will be replicated. So one of the interesting things that this drug does when you're giving it to people with CLL, which is actually a little worrisome if you don't understand it, is that it causes the white cell count to go up, okay? And normally when you're treating CLL, what happens? The white cell count goes down. And so if it's not going down and it's going up, what's your normal conclusion? Is that the drug isn't working, right? And what they had seen was that it didn't make sense to think that the drug wasn't working because people's lymph nodes were shrinking, but their white cell count was going up. And so they thought, is this some bizarre phenomenon that the drug is doing? And what they actually showed was that this was a mobilization phenomenon, perhaps of cancer cells leaving the bone marrow or leaving the lymph node or leaving the spleen and getting into the bloodstream and going up. And then what you see over time, so here are the size of the lymph nodes. The lymph nodes impressively come down and continue to come down over months. But the white cell count, the lymphocyte count went up, peaked by about the second month, and then started to come back down again as well. And so that makes it a little difficult to interpret some of the data because, again, the way we define response, lymph nodes go down, that's good. Counts in the blood go up, that's bad, and we wouldn't have actually called that response. So you may see some funny numbers to sort of describe that here, actually right here. And so the other interesting thing, this type of study, so again, this isn't a phase 3 study, this isn't even a phase 2 study, this was a phase 1 study. So this is the first time that people got this drug, but the drug looked so promising in a phase 1 study, they continued to accrue some patients, so it's a large phase 1 study. Here we're looking at over 80 patients, and it was so effective, it changed practice in the US based on these sorts of results, with no control arm, no comparison against a standard of care. And it's because the results look pretty impressive. And you can see here, the percentage of people that responded was actually 70%, okay? And if you add in the people who had the response in their lymph nodes, but you saw the white cell count go up initially, that's another 20% of the patients. So it looks like a lot of people are benefiting from the drug. And in comparison, they looked at 2 doses. So again, they were still trying to figure out what the right dose of the drug was in this phase 1 study. You could give double the dose, 840 milligrams or 420, and it worked just as well. Okay, so financially that might be reassuring because you can get away with a lower dose of the drug. And so again, this is a pill that's taken every day, and you stay on it. It doesn't stop at 3 months or 6 months. You stay on until you progress. So the theory here, it's like the daily vitamin. Okay, you're on it until it stops working. How did it work? So just very shortly, this looks at genetics in the CLL cells that predict for outcome. And normally, this group of patients here, those with 17p deletions, this is the hardest type of CLL to treat. And what this study shows is that this drug didn't care, okay? It worked equally well, essentially, even though that curve looks different. They don't have enough numbers to show that it's worse than this. It looks like it probably is, but there's no difference. Again, it didn't hit that magical number of 0.05. But what you can see is that the drug was effective in all patient populations in that study. And looking at another marker, immunoglobulin gene rearrangements, where again, one group should do much worse than the other, the drug was agnostic. It worked incredibly well. And this is the sort of thing you want to see about a drug that could potentially change practice, is that it works in the worst group of patients. So when they looked at this in mantle cell lymphoma, again, they gave a slightly different dose. It was a little bit higher, 560 milligrams. But this was actually a phase 2 study where they were looking specifically at this disease. And they looked at 2 different groups of patients. So again, in the US, Velcade or bortezomib is a standard treatment to give in mantle cell lymphoma. And so they looked at a group of patients that had had that before and a group of patients that didn't have it. And again, this was a single-arm study, so it wasn't being compared against a standard. And what happened here, again, it didn't matter if you'd seen one of the other standard novel drugs in mantle cell lymphoma. Ibrutinib was just as good in both patient populations. And ibrutinib looked very active. And again, in terms of staying in remission, not just survival, it also looked to be just as good. So the difficult thing in terms of interpreting those is, do you know how good it is compared to something else? You don't, okay? Does it look useful? For sure. And there are now a whole host of new studies that are kind of coming to fruition right now. And there was one that was actually just published in the Journal of Clinical Oncology earlier this week, where they compared ibrutinib in relapsed CLL against ofatumumab, which was that antibody I had shown you previously in CLL. And this was yet another treatment, now the 4th treatment. We'll talk about the 3rd in a second. But the 4th treatment to show an improvement in overall survival in CLL in the relapse, and this time in the relapse setting. So ibrutinib, they went on to do the confirmatory phase 3 study, and they showed that it was better than ofatumumab in the relapse setting. What we're excited about in Canada is the opportunity to participate in a bunch of frontline studies with this drug as well, and those are now rolling out to studies across the country. So this is a subset of the pathway, again, of the B-cell receptor signaling pathway. And I referred to it earlier, something called PI3 kinase, or PI3K. And the interesting thing about this drug, which is a drug called idelalisib, is that it was designed to affect something called PI3 kinase delta. And so this was the idea here. With a targeted treatment, you want to be as targeted as you can. And ideally, if the target is only in a cancer cell, so unlike rituximab, which affects normal B cells as well, if it's only in the cancer cell, you can be very selective about what you affect in terms of protecting healthy cells and affecting cancer cells. So a truly targeted principle here. And so this is that point that this is restricted to white cells, so not to cancer cells specifically, but to white cells, but sort of showing that you would expect less side effects with this drug if it was more broad in its application to the various types of PI3K enzymes that are out there. Okay? So this was a study that was again presented back at the American Society of Hematology meeting in December and was then published fairly soon after in the New England Journal of Medicine. And what they looked here was again older CLL in the relapse setting and patients that were not felt to be eligible to get more aggressive chemotherapy. And so the comparison was against rituximab by itself in CLL. And the experimental arm was to give this drug idelalisib, which is the PI3 kinase inhibitor, in combination with rituximab, where the rituximab was given for a finite period, 6 months. And like I told you with ibrutinib, this is a drug that you stay on treatment indefinitely as a pill until the drug, potentially until progression. And what they showed here was that at progression, the opportunity was there to get the drug at a higher dose if you were already on it, or to be crossed over. Okay, and so if you didn't get the drug, the chance to get the benefit from getting it. And so you can imagine then, in terms of survival, this trial wasn't powered to look at survival because they were going to give the drug in both arms eventually. It was designed to look at that, how long can you stay in remission or how long until the disease grows. But what they saw was surprising, which was that even though the trial, you could argue, was built against this from happening, this was drug number 3 that shows an improvement in overall survival in patients with CLL compared to rituximab. Okay, so the combination of this drug plus rituximab is better than rituximab alone. Okay, and that looks fairly convincing. So that was a very good result in that relapsed population, patients who wouldn't be eligible for chemotherapy. The interpretation, the thing that makes this one a little more tricky for us, is that if you talk to a bunch of people that treat CLL in Canada, do we use rituximab as a single treatment by itself in relapsed CLL? And the answer is no, we don't. And why is that? It's because, as I told you a little while ago, rituximab by itself is actually not a very good treatment in CLL. Okay, and so this is one of those interpretation questions where in Canada we will say, you did a comparison against a standard that we didn't think was really a good standard. There's no doubt the drug is better than rituximab alone when it's given with rituximab. But, you know, is the reason that this looked better just because what you compared it to was not very good in this setting? And that's the important piece here. What we learned from our colleagues in the US was actually rituximab is used quite commonly as a single treatment by itself in the US. You know, they can do that. It's not based on a lot of evidence. And so it's an open question. And so I think one of the things that will make it a little more difficult for this drug to see approval and be funded in Canada is that very issue, because in Canada we'll have to say, how would our standard have looked in the second-line setting against this combination with rituximab, and is it really better? You know, again, what I told you before was that we didn't have very good treatments in the relapse setting in CLL either. So, you know, I believe the drug clearly has a role. It just may be a little more difficult to try and make the argument based on that study. And just to briefly mention, they've done a very good study similar to the ofatumumab study where they took this drug and gave it in a very bad group of patients with refractory follicular lymphoma. The drug had significant activity there. Again, almost analogous to the study I showed you with ofatumumab. And so proof of concept, it works as a single agent in very bad follicular lymphoma. We would like to give it much earlier in the disease course, and we'll just need to see how those studies play out over the next little while. But stay tuned. As I said, this pathway, these targets, these drugs have a much broader role outside of CLL. Okay, so we're going to switch gears and talk about a drug that's a little older now, but remains an emerging target in lymphoma, a drug called lenalidomide. And so those of you who are familiar with a disease called multiple myeloma will know this is a very standard treatment in multiple myeloma. Again, it's a pill that you normally take 3 weeks out of every 4. I'm always worried to show slides like this, where the mechanism is broad, because this doesn't speak to saying that we really understand what it does. Beyond it does a whole host of things that are very useful. It may be directly toxic against cancer cells. It may do things that may relate to the blood supply to tumors. It may do things specifically against multiple myeloma cells. It certainly changes the immune profile, and it may supercharge the immune system, and that may be part of the mechanism of how it works. But I will say it is a complicated drug, and much remains to be sorted out. So this is an example of one of the studies that's been done with lenalidomide that was published recently, and much like the trial with ibrutinib, this was done in relapsed refractory mantle cell lymphoma, but here in this study, everyone had had bortezomib or Velcade before. Much like the other study, not a comparison against another standard treatment, this is just looking at the drug by itself, and you can see here, it has some activity at about 4 months in terms of keeping people in remission, but that doesn't look like that promising a curve. It shows that the drug works, but one of the struggles with mantle cell lymphoma is once you get beyond 2 or 3 treatments, it's a very difficult disease to treat. And if you can remember what the curve looked like for ibrutinib, did it look better or worse? This is where it gets very dangerous about comparing small numbers of patients and studies between them. So I'm not going to say anything more than that. The survival, the median here was about 1.5 years, and that looks okay. It looks better than what you would accomplish with standard drugs in this setting, but again, not compared against the standard. So one of the other areas where this drug looks very promising is as a frontline treatment in follicular lymphoma. And so Nathan Fowler, who's a good young investigator at the MD Anderson Cancer Center in Houston, led a study that looked at giving lenalidomide in combination with rituximab as the first treatment for a symptomatic follicular lymphoma. So if you think about that for a second, this is now a combination that has no chemotherapy in it at all. Okay, so we decided bendamustine is maybe the best chemo drug, but here, this is actually testing a whole new concept. Can you get beyond chemotherapy whatsoever and just give targeted treatment? If you can say we understand how lenalidomide works. But anyway, it's a proof of concept thing that looks great, and it has led to this study, which is, has this moniker of relevance, and it's being run also through the NCI Canada, the National Cancer Institute of Canada Clinical Trials Group, where they're taking 1,000 patients with newly diagnosed follicular lymphoma that need treatment, and they're going to get Revlimid plus rituximab, or they're going to get rituximab in combination with chemo, whatever people would give, and in Canada that would be bendamustine. And then following that, they go on to get rituximab and lenalidomide as maintenance, or just get rituximab maintenance by itself, with the idea here to show proof of concept that you can improve outcome for patients by giving a regimen that is entirely chemo-free. So that sounds very exciting, because what did we start off with? The idea that chemotherapy didn't make a big difference. Previously, bendamustine appeared to make a difference, and now can we entirely get away from it? So I think it's a concept that we need to really try and understand. It's very appealing. I'm not saying that we can throw away chemotherapy entirely, but this may allow us to reserve chemotherapy for down the road, or potentially for a subset of people that may need it more. Or we give this type of treatment to people that a drug that affects immunity and a host of other things might be very relevant for. Relevant for relevance. There you go. So again, lots of promising activity in a number of diseases. I've just shown you a couple of snapshots, as well as with large cell lymphoma. A bunch of trials are waiting. I will mention they did a trial in older patients with CLL where they saw problems with this drug. And despite it being an approved drug and myeloma being used in tons of places, we know that there is potentially a downside with all of the drugs that we give. And what's the downside with lenalidomide? They have shown that there are risks, just like with standard chemotherapy, of people developing second cancers. That risk may be increased in people that are getting lenalidomide as maintenance. So that's an important issue as we look to this in diseases like CLL and follicular lymphoma, where people may live for a number of years. The other issue here in this older CLL study is that there was some increased toxicity and death. Okay, and that's why we don't feel comfortable just kind of rolling this out and say, oh, it looks good, MD Anderson did this, let's just give this to you as a frontline treatment, because we may see things in these studies that will influence that. The problem with the study I showed you is it will realistically take 10 years to actually get the answer. And that's because it's 1,000 patients. The standard arm is so good to show that it's going to be better, it will take time. Okay, so we're going to talk about a drug called romidepsin now. And so this is one of the drugs that represents a different class of agent called an HDAC inhibitor. That stands for a histone deacetylase inhibitor. And this was approved a number of years ago by the FDA for patients that have a rarer type of lymphoma, cutaneous T-cell lymphoma, and approved a couple of years later for patients with, again, a more common type of T-cell lymphoma, but still rare, something called peripheral T-cell lymphoma. What is the principle here? HDAC inhibitors inhibit this enzyme. And what this does is it actually does something epigenetically. So again, this is another concept that's very important in cancer medicine in general. The idea here that if you inhibit this drug, you keep these genes and this chromatin coiled and closed, and that prevents those genes from turning on to produce their product. So if you have a cancer gene and you can turn it off, as an example, you may actually prevent it from doing all the bad things it's supposed to do. So that's the concept. Harder to deliver in practice maybe, but what they showed, so relapse refractory peripheral T-cell lymphoma is a very challenging disease to treat. And this is a group of patients that might have failed stem cell transplants or were too old to have had them. And so this was actually quite a positive finding in terms of people staying in remission after the drug. The appeal, and the thing that we need to understand better, is this group of patients that look to be doing well and staying on the drug for more than a year or 18 months out. And so again, what is different about those patients or their lymphoma that they respond so well to a genetic type treatment? And that's something we'll need to see over time. Again, as a quick reminder, this is just how the patient group looked. They were a variety of different people. Most of them had had at least 2 or more treatments. Half of them were refractory to the last treatment they had, so a very tough group of patients to look after. And how well did it work just in terms of the activity? So about 30% was the chance that you respond to the drug. And again, you'll remember the bar that I mentioned with rituximab from the very beginning was very high at about 50%. For most drug developers, if you see that the drug causes the cancer to shrink 20% of the time in people that have failed multiple treatments, that's a sign that you have potentially a very good drug to work with. All of the data I've shown you up until now have been largely home run drugs, and that's because they work in 70% of the patients that they see. So for most cancers, we don't see that. And so we've been lucky in lymphoma and CLL to have had these types of drugs available to us. The toxicity with all of this class of drug is actually the same. It's tough on blood counts, particularly platelets, and it's potentially a little tough on the GI system in terms of mild nausea or vomiting. So I can't say it's necessarily better than combination chemotherapy in that regard. It's probably similar in terms of its side effects. So what's happening with romidepsin? So it's now been approved by Health Canada for use in Canada. Again, the question here might be where the funding is going to play out at the provincial level as we move forward with getting that looked after. But what we try to do with these drugs when we see that they work, again, as you've seen, is to move them into combination or to move them earlier. And again, a group in France is combining it with CHOP to kind of see how it does as a frontline treatment. And what we're doing in Canada is actually being, maybe even a little more forward thinking to say that we're going to give it with a regimen that we think is very good in T-cell lymphoma, gemcitabine, dexamethasone, cisplatin, which is a Canadian regimen. And if that looks good, we may try to replace CHOP with this in T-cell lymphoma, because there's certainly room to improve the outcome in that disease. So we're going to go back to antibodies for a second here, because we've switched diseases again. And this is to talk about a very interesting drug called Adcetris, or brentuximab vedotin. And this builds on the concept that I told you about rituximab from the very beginning. Okay, so this is an antibody, but the difference here is the antibody has a payload. And so by that I mean they've attached chemotherapy to the antibody, which makes this a class of drug called an ADC, an antibody drug conjugate. And so the chemists are, the difficult thing to do with this is to actually figure out this piece, how to attach the drug to the antibody so that it does what you want it to do. And what do you want it to do? Well, you want it to do this. So you want it to still attach to the target on the cell, and in this case, it's not CD20 like with rituximab, it's CD30, which is another cell surface protein on lymphoma cells. But then it gets taken in, and then the art of all of this was that linker that attaches the chemo to the antibody gets dissolved in the cancer cell, and then the chemotherapy is free to target and wreak havoc in the cancer cell it just came into, right? So what is the appeal of this? You should see very little side effects outside of the cancer because all of the drug is being designed to be homed in on and delivered to the cancer cell. So again, the principle here is great. This is the poster child example. And in what disease was it? So CD30 is a target people have looked at in Hodgkin's lymphoma for a long time. And this was the first drug to actually target that as an antibody. And so the study that got a lot of excitement was this trial, which was a large phase 2 study in people with Hodgkin's lymphoma who have all failed 2 types of treatment. They've had a stem cell transplant and had the disease come back after that. And so how is this drug given? It's given once every 3 weeks, intravenously, over 30 minutes, and it was potentially to give up to 16 treatments. So if you do the math, that's a little under a year. It's 48 weeks of treatment. So the problem with Hodgkin's lymphoma is that these are the types of patients that you look after. And so the median age was just over 30, and there was a fairly wide spread there. And just to highlight here that the majority of the people, so as many of you know, the majority of patients with Hodgkin's lymphoma have a success story. They are cured, they're young, they do very well, but we spend a lot of time dealing with difficult Hodgkin's cases because when they're refractory, they're potentially very refractory, like aggressive non-Hodgkin's lymphomas. And this was a very bad group of patients in that about 3/4 of them had had their disease come back within a year after their transplant. So this is probably the toughest group of Hodgkin's lymphoma to treat. And again, what did I tell you the bar was? 20% is a minimum, 50% for a superstar drug. The response rate here, 75%. Okay, 1/3 of the patients went into complete remission from getting this drug. And very few, you can see here, 3% actually grew or progressed on the treatment. So it looked very, very effective. So this is maybe the reason to hesitate a little bit about this drug. And that's because though 3/4 of the patients had a good response, the time in remission was not very long. And so it was only 6 months, 5.5 months. And so again, why? It's probably because it's being used way too far down the road in a very difficult group of patients, as I said. But still, compared to some of the other things we were giving, likely better, but again, it wasn't compared in this setting against a standard treatment. What they tried to do, and so this is another one of the tricks that investigators do or companies do to try and get at this in a non-randomized study. So they said, all these patients had a treatment before this. How did this do compared to the treatment they got before? With the idea that certainly you wouldn't expect the next treatment to be better after just failing something else. And what they did show was that this actually statistically did better than what you got with the last treatment, suggesting that maybe there is proof of concept that, again, this is another game-changing treatment. How was it from a toxicity standpoint? So the main issue with this drug is, or issues are these two. So one being sensory neuropathy, so numbness or tingling in your hands and your feet that can get progressive. And so grade 3 or 4 is significant. It's the sort of thing that we might consider modifying the dose or potentially stopping drug. Grade 3 or 4 neutropenia in 20% of patients. And again, needs to be taken with some interpretation, right? Because these are people that have had 2 or 3 different treatments at least. Often we use classes of drugs that can cause nerve injury with first-line and second-line treatment. So the context is it's not entirely innocuous, but it's certainly a drug that is safe, but you have to learn how to use it. So what happened, so this got approved in the US last year. It is now finally approved and funded in Canada. And so patients are now finally able to get this as a standard treatment in Ontario. It's taken a long time. So yeah, sorry I didn't update this slide, but yes, it is now approved and funded. And what are we doing in this scenario? So people are looking at it in maintenance after a stem cell transplant to prevent recurrence. And the most important thing is it's being tested in the frontline treatment setting. So it's being combined with ABVD, essentially, to try and show that it's better. And again, proof of concept that you can take an antibody drug conjugate. It's the first time this was really shown in cancer, and actually make a big change in terms of outcome. And we believe, of course, we believe that this study will be a positive one, but we'll need to see after several years if that's going to be the case. So that was kind of a whirlwind tour about new treatments in lymphoma, non-Hodgkin's lymphoma, Hodgkin's lymphoma, and CLL. And as you can tell, I think, I'm not the only one that is excited about the fact that we have a lot of new things to be able to offer people, because you can see the drugs are active. A lot of the drugs are oral. A lot of the drugs don't have a lot of side effects. The issue, and where the advocacy piece becomes critical in all of this, nothing in modern healthcare comes cheap, right? And so the cost of these drugs, these oral drugs like ibrutinib, but as an example, cost about $100,000 US per year. And it's not you get 6 months or 12 months and you stop. You stay on until you progress. And our worry, right, like as a society, has to be, can you absorb that type of cost for common diseases? It'd be one thing if this was 5 patients in Canada, but CLL, follicular lymphoma, large cell lymphoma, like these are very common diseases. We're number 5 or 6 in incidence in Canada, depending who you talk to. And that's not to say that's great, lymphoma is common, but it's going to be an issue because the public purse supports this in Canada, and how are we going to make all of this happen? And so that's where advocacy will be important. That's where hopefully clever people who know how to look after people can help impact on where we may use these drugs most effectively, because it's going to be a very, very difficult thing moving forward. And so in that regard, for people that look after patients, and for patients, the idea here is we will try to do all we can to ensure that people have access to these drugs through clinical trials, firstly to try and continue to prove that these sorts of drugs are actually helpful and useful. And again, as a community, we have to continue to advocate for treatments for lymphoma patients, specifically in Canada. And that's what we hope, obviously, as you know, we hope to do with Lymphoma Canada. And so thank you, and I can take some questions, but that's probably been quite a long time to listen to me at any one time. Okay, thank you.
Thank you very much.
I'm 62 years old, DLBCL lymphoma, 3 years in remission. My question is there any maintenance drug for this disease? And are there any trials?
Sure, thank you. So maintenance, I'll answer it a little broadly, I think. So maintenance therapy in lymphoma has been shown to be of benefit in follicular lymphoma. And so as a standard treatment, we've actually not been able to show that maintenance was successful in large cell lymphoma. And the drugs that have been tried to date, they include rituximab was actually tested in large cell lymphoma as maintenance, both in the primary and second treatment setting. Didn't work. A drug called Avastin, an anti-angiogenic drug, also was tried and didn't work. A newer drug called enzastorin was also tried and didn't work. And we're waiting to hear the readout on a couple of studies. One's with a drug called everolimus, and the other one is with a drug called lenalidomide. So we're hoping to get results of those studies, sometime in the next couple of years. I guess to answer your point specifically, for someone who's been in remission for 3 years, the way that these studies have largely been done is the maintenance is not given indefinitely. It's usually given for a period of a year or 2 years after the completion of primary treatment, because thankfully, the highest period of risk with this disease is usually within the first 12 to 24 months. So, you're actually through the worst window, and getting additional treatment at 3, 4, or 5 years, first is unlikely to help, but the studies generally haven't continued treatment for that long.
First of all, Dr. Kuruvilla, thank you very much for your most interesting presentation. I look forward to reviewing it. There's a lot of information that you provided to us, but my question really has to do with a topic that is central to follicular lymphoma and perhaps chronic lymphocytic leukemia as well, and that's Richter's transformation.
Mm-hmm.
1 out of every 3 patients with follicular lymphoma is at risk of having a transformation. And this is a pretty tough lymphoma to treat, and I'm sure you know what the stats are on it. It's pretty grim. I was excited to hear you talk about the studies that were being done by Nathan Fowler and others trying to dump chemotherapy from all forms of treatment with follicular lymphoma and move into a field where you're dealing with drugs such as Lenalidomide and Rituximab alone, so that, because it appears that from the little that I've read, there's pretty strong evidence emerging that if you have chemotherapy, particularly in frontline treatment of follicular lymphoma, there seems to be a significantly higher risk of Richter's lymphoma transformation occurring. It's one of the driving forces, as I understand it, toward trying to get rid of the chemotherapy in frontline treatment. In other words, even though R-CHOP was good in its day, and rituximab bendamustine is filling in underneath that, if we can develop a system of treating follicular lymphoma frontline without any chemotherapy, it could alleviate the difficulty with Richter's lymphoma. And given the fact we don't have treatment for curative intent with follicular lymphoma at this stage, it seems as though that's a pretty good idea. Especially for somebody like me who sits here at, what am I, grade 1, 2, stage 4 follicular lymphoma, still asymptomatic after almost two years. So I'm concerned about that, because at some stage if I do need systemic treatment, I'd like to know that it's the best. Mm-hmm. Do we have clinical trials in Canada that deal with, or are we associated in clinical trials in Canada that deal with engaging non-chemotherapeutic agents for frontline treatment of follicular lymphoma?
Definitely, yeah.
Okay.
So I guess I'll answer that part of your question first. So the study that I showed there that looked at the combination of lenalidomide and rituximab versus chemotherapy with rituximab is being run in Canada by the NCI Canada, and so it's available at most major centers across the country, and so certainly in the GTA, Sunnybrook, ourselves, a number of other centers will be running it, and it's available. To answer the other question, I think what I'll say, so recently, for whatever reason, a lot of my research has actually been focused on transformed lymphoma, and I think there are reasons to be encouraged. And so firstly, if you look at the outcome of transformed lymphoma today compared to what was published 10 years ago, the outcome is night and day better. Why is that? I have to admit, I don't think it's because that the doctors have figured out how to treat this disease any better. We haven't gotten any smarter, I don't think. But the one big change has been rituximab. And so if you look at the results that are now coming out from the groups that have reported about this recently, if you get R-CHOP for your primary treatment of transformed follicular lymphoma, your outcome is just as good as getting treated for large cell lymphoma with R-CHOP in the beginning, okay? And so that's a huge point, because we appear to have equalized the outcome. The reason transformed lymphoma historically was felt to have done so poorly because it was often viewed as an end-stage event. And so in the days when we would have exhausted chlorambucil and CDP and CHOP and what have you, you had A, little option, and B, certainly nothing innovative in terms of what you could have available. The driver in that disease, again, is going to come from the lab. It's going to be translationally identified as being tumor genetics, essentially. And so in people that have had aggressive treatment, or people that required treatment for their follicular lymphoma previously and may not be able to get R-CHOP as the treatment if they've had it before, as an example, what we've done in that scenario is we recommended people that are well enough and fit enough up to the age of 70 to get different chemotherapy and then to have an autologous stem cell transplant, one that they donate stem cells to themselves. And again, the data that's come out largely from Canada, led by our group, actually shows that, again, you do just as well as patients who've had transplants for regular large cell lymphoma. So while that's not a home run, at least half the patients that may have those transplants may be cured of the aggressive lymphoma. Beyond that, where things are going to move in that area will certainly be with more targeted drugs. And so this is an area that the biology of lymphoma is being studied in many different places, and the genetics of transformed follicular lymphoma, not surprisingly, have some unique targets. And so that's where people are going to be focusing on. And we have a drug in the clinic, actually, in a few centers in Canada and across the world, where the plan is actually to take that drug forward specifically in patients with transformed lymphoma, particularly in CLL that has transformed large cell lymphoma. That's what you call Richter's transformation. Richter's only applies to that term within CLL. But this drug was felt to look as promising in that group of patients that they're going to do a specific study in that rare type of patient. Okay, so lots of reasons to be encouraged because the platform's gotten better already and people are now moving in with targeted treatments in that setting.
We do have a number of questions coming in from those watching our webcast, as well as our group in Waterloo. And I did want to mention to everyone in this room, we are filming this. However, you will not be filmed if you asked a question, only your voice for your privacy. So here's a question out of Waterloo, Dr. Kuruvilla. And it is there a gap between a new treatment's use in the larger cancer centers and their use as standards of care? Meaning, are we more likely to get access if we are being treated in a larger center than otherwise?
Okay, that's a very good question. So, the easiest way to answer that is before a drug becomes approved and funded and available, the only way you can access the drug is in the setting of a research study in a clinical trial. And so, again, in a smaller program, firstly with fewer resources, fewer personnel to run clinical trials and run infrastructure, they will have fewer clinical trials, right? So that means naturally larger centers will have those trials available, particularly if you have a very rare kind of lymphoma. So if you have relapsed Hodgkin's lymphoma or peripheral T-cell lymphoma, there are a handful of centers in Canada or even in Ontario that may open trials for that rarer population. So the business end of this is that trials cost money to run. You take on a lot of effort to be able to run a trial in a center, and so to open a trial for one patient, while that sounds emotionally very appealing, it never works. And that's where, again, the advantage of what we have in Canada and Ontario is you have the network, right? So we frequently see people, from Kitchener or from Hamilton or wherever about trials that we have open that would be relevant to them. Where the bigger gap, unfortunately, grows as the distance grows, right? And so if you live in Northern Ontario, your access to clinical trials is a problem, right? And so unfortunately, that does lead to some inequity in terms of access to research studies. There isn't a very good answer for that because at the end of the day, people who go on to clinical trials, these are experiments, right? There is a lot of safety that is built into how studies are conducted. You are held to a very high bar in terms of what happens, how you perform a study, how you may be audited, what happens if something goes wrong. And it becomes very difficult for small groups to do that, or for us to be able to be willing to say, we can manage someone from Sault Ste. Marie on a clinical trial where you need to be seen weekly and where I need to know exactly what happens if you develop a side effect. It can't be you're getting standard treatment, you can phone me, we'll figure out what to do. I may need to see you in 6 to 12 hours, and that becomes, not theoretically impossible, it just becomes practically not feasible.
Thank you. We have a question for Margot. She has 3 family members, including herself, that have indolent follicular lymphoma. And she's wondering, are there any studies on prevention as opposed to treatment?
So that's a very good question. So there are no studies examining prevention in lymphoma at the moment because unfortunately we don't really understand the mechanism of the disease well enough to understand how to prevent it. So the epidemiology here is very interesting, right? There are descriptions with most cancers that you may see clusters within families, right? But does that mean that there is a true inherited genetic basis to get lymphoma? Not that anyone's clearly proven. So unlike breast cancer, where there's a story with a cancer gene, BRCA1, that you can see transmitted in families, we don't have that story in lymphoma yet. We also don't have an etiologic factor per se, so you can't say like lung cancer with smoking, cigarette smoke and lung cancer that there's a link. So we can't say that about lymphoma yet. All we know about lymphoma in terms of why it happens in broad strokes, we see it more commonly as the population ages, though there are some diseases like Hodgkin's lymphoma, again, where there may be a peak in a youthful era, and the distributions of these lymphomas may vary a bit by age. We know that certain things put people at high risk, and not surprisingly, with lymphocytes being part of the immune system, immune diseases may be part of the predisposition, right? So HIV is an example of an immune disease where you see higher rates of lymphoma. Patients who've had solid organ transplants, so heart, kidney, liver transplants, are at higher risk of developing lymphomas, again, likely from the drugs that they're given to suppress the immune system. And now for autoimmune diseases, lupus, rheumatoid arthritis, Crohn's disease, the list goes on. The fancy immune drugs that they're getting, antibodies like rituximab in a way that target parts of the immune system, that puts them at very high risk, or well, not very high risk, but puts them at risk of developing lymphoma as a consequence. But we don't understand much more than that. And I have to admit, this is an area where the research community needs to do better. The appeal of drug studies is naturally because it's in the patients that you treat and pharma is vested, but understanding the why is probably ultimately the more important goal, because I agree, it's far better to prevent a lymphoma than treat it, but we don't know how yet.
Thank you. Another question we have from Patrick. He's wondering when some of these drugs will be available, specifically the ibrutinib and idelalisib.
I don't know. So this is something that's happening in real time, right? So we know that that there's a pathway to how drugs get approved. It does involve approval nationally with Health Canada, which sort of speaks to some safety and efficacy. They don't bother to comment on pricing and cost effectiveness, those buzzwords that are out there now. And that's done by another group now nationally. And so that process takes months, it does, and can take a lot longer. Unfortunately, it's always longer than what we would like. So some of the companies do have the opportunity to step in there with an access program of some sort, though that seems to be increasingly fraught with some challenges. But I know these drugs, these companies are actively engaged in trying to move forward, and we hope they come soon. But I've often thought I knew that a drug was coming in very close, and then 6 months later, we're still waiting to hear, because sometimes the bureaucratic wheels, as you know, in Canada, turn very slowly.
Any questions in the room?
Hi. You've talked a lot about different types of lymphomas, but you didn't touch greatly on diffuse large cell lymphoma. And as far as I'm aware, being new to this subject, R-CHOP is the gold standard at the moment. Is there anything on the immediate horizon in terms of treatments, antibody drugs, to modify that R-CHOP treatment?
Right, so I did speak a bit to that in the beginning, right? So the drug obinutuzumab, the second generation rituximab, the new anti-CD20, is actually being tested frontline in large cell lymphoma now. The answer about maintenance, again, a number of trials have been done looking at giving maintenance after primary treatment with R-CHOP for large cell lymphoma. There are a number of trials open actually looking at large cell lymphoma. So combining lenalidomide with R-CHOP is something that's being done now in a big frontline trial. Bortezomib, so that drug Velcade we talked about in relationship to mantle cell lymphoma, a large group in the UK actually running that study, looking at it in combination with R-CHOP. Part of why that shopping list of specific drugs was put together was, I think that was based on feedback that was given to Robin and Lymphoma Canada about what you wanted to hear about. And so, I could have done 90 minutes on new drug development in large cell lymphoma. That's actually my main area of interest. But it's hard. Large cell lymphoma is probably the most challenging disease to develop drugs in, because it's biologically, it's a very heterogeneous disease. There are a lot of genes, there's a lot going on. And once you fail primary treatment or you fail a transplant, it's a very difficult disease to treat. And unlike CLL or follicular lymphoma, where there's sometimes more time for drugs to work because the disease is a little more slow in how it progresses, large cell, as it is characterized as the most common aggressive lymphoma, can behave aggressively. And so drugs are put to a very high standard in that setting to see that they work. It's a challenge, but there are other encouraging drugs that are now in early phase studies there as well.
Any other questions from the room? We have lots of questions coming online, also from Waterloo. Peter from Ottawa asks, where is the best place to find information about clinical trials upcoming in Ontario?
So probably the best answer about where to find clinical trial information, if it's really about understanding a drug or a disease, is actually to look on the NCI's website, clinicaltrials.gov. So it was designed to have both a patient and a professional area, and it will provide a list. You can search it by disease or by drug and sort of see what's out there. And we're all sort of mandated to provide information to those studies, to that site, because all clinical trials, if they want to get published, need to be registered. Lymphoma Canada does have some resource available in that regard and is being developed. All of the trials groups that do trials in Canada tend to advertise to the medical community. Okay, so as an example, we send out a list of our trials monthly, likely to all of the treating physicians that would be involved treating patients who are local to Ontario anyway. And so people know what we have. So I would say in general, the other best option is to check with the team that's actually looking after you, because they will know, as an example, what we're doing, what Sunnybrook's doing, any of the other big trial centers, Ottawa, Hamilton, etc., as to what's going on.
Thank you. We do have some questions related to advocacy, but we'll save that for part 2. This is a question about stem cell treatment, and it is from the Waterloo group. When is it administered alone, or can it be administered with other drug treatments, as you mentioned in your presentation, the other drugs that are upcoming?
Yeah, so that really depends on the level of evidence in terms of what you incorporate into the transplant. So we spent 10 years doing a study looking at second-line treatment in transplant, where we were trying to figure out the chemotherapy before the transplant, and then we were looking at maintenance after the transplant with rituximab. So until one of those treatments actually improves outcome, that will remain in the place of clinical trials, essentially.
Okay, and another question we have is, what do you think is the most promising area of study for new treatments?
So based on what we have at the moment, I think the most appealing thing, just because of the breadth of agents that are available and the fact we've already seen success, probably relates to the story around the B-cell receptor. The other area that I'm quite excited in is the principles around anti-CD20, because we know that's already changed outcome, and you can speculate now that we may change outcome again for the better because there's a newer agent that's available that looks more promising. The tough thing in all of this, though, is that lymphoma in general is a disease of many, right? So the biology is complex. We've already talked about many different subtypes of lymphoma. So I think in broad strokes, the answers are going to be, you want to look at targets that affect the cancer broadly, right? And that's where, looking at things like PI3 kinase, or looking at a target that's on all B-cell lymphomas, or again, modifying epigenetics with an HDAC inhibitor, that's where I think the money is going to be, is that despite the idea that you can have one gene and one treatment, lymphoma has many genes and many different types of lymphoma, and I think that the approach is going to have to be a little more sophisticated.